Students, today we're going to take class on polysaccharides. Maximum in this video, I'm going to cover total biomolecules. So here, this all will be, I will keep in a playlist. If you want, you can, at a stretch, you can see that all videos. So here, biomolecules, polysaccharides. Today's concept is the polysaccharides. These are polymers of sugars. Polymers of sugars means what? No, monosaccharides. Number of monosaccharides as a polymers, right? Example: starch is the one polysaccharide, and cellulose is the one polysaccharide, and glycogen and inulin. These are the polysaccharides. What is the nature of starch? This is the homopolymer of glucose, whereas the cellulose is the homopolymer of glucose, same, and this also homopolymer of glucose, but inulin is the fructose. Okay, fructose. Inulin is the, see here, this is the one kind of protein, means polysaccharide. It is the homopolymer of fructose, same kind of fructose there. Here same kind of glucose there, and here also cellulose have a number of glucoses there. These are the what you call polysaccharides, right? Here is, see here, there are complex polysaccharides formed of amino sugars, which are the glucosamine and N-acetyl galactosamine. These two are the complex polysaccharide examples. What are the examples of complex polysaccharides? Which are the glucosamine and N-acetyl galactosamine, right? One more is the chitin. Chitin is the homopolymer of N-acetyl glucosamine. It is seen in the exoskeleton of arthropods. We know that arthropods exoskeleton is made up of the chitin and fungal cell wall also. This is very important. Fungal cell wall also, which is composed of chitin. See, in the detritus, if chitin is there, the decomposition process will be slower, right? See, glycoprotein, amorphous B glucan and B glucan, these all are the fungal cell wall is the main composition, which is the chitin, right? They have given as a some crab and fungi, right? Next, what is the glycosidic bond in the polysaccharides? Polysaccharides have a glycosidic bond. Here is the monosaccharide subunit is there here, and disaccharide. Here is the disaccharide. Between these two monosaccharide subunits have a bond is there, we call it as a glycosidic bond. It is the bond formed when individual monosaccharides are linked between the two carbon atoms by the dehydration. Because of the dehydration, two individuals, this is the one and this is the other one, these two monosaccharides are linked with the carbon atoms. See, this is the carbon atom, here is the carbon atom, here bond is hydration H2O, hydration takes place now. Here is the OH, here is the OH is there. Now oxygen is there, H2O, hydration takes place. That bond is called as a glycosidic bond, right? Now here is the diagrammatic representation of the protein glycogen. Sorry, this is the glycogen. Glycogen is the protein that actually is visible like this, right? And here is the starch forms the helical secondary structure. In this starch, it can hold iodine. You know that presence of starch, you're using the what you call experiment, that is the iodine test. Cellulose has a no complex helix, therefore cannot be hold H2. Why starch is holding iodine? Because it have helical protein, that helical portion giving the blue color. But cellulose, why should not be holds the iodine? It does not have any helix, right? Iodine test often using for the presence of starch, right? And here is the next. Nucleic acids, this is a very very important one. Nucleic acid, DNA and RNA, we will discuss this in the next video. Today onwards we have to upload every day one video until completion of the biomolecules. Thank you students. Today we're going to take class on poly